Hey everybody, I hope you had a good weekend. So I'm going to teach you about mutations today, which is really fun. So some things that you should know, just some vocab that you should definitely know before starting this lecture. You should know what a codon is. You should know what a base is, A, C, G, and T. You should know what a gene is, DNA, mRNA, protein synthesis, amino acids, ribosome, and nucleus. So you should be familiar with these words. If you're not, I think you've missed a few of my lectures, so you might want to go back to past assignments if this is very confusing to you. Just for some extra review, I'm going to show you this protein synthesis kind of visual, and I'm going to talk you through what you're seeing. So this is what we should have learned up until now. So the first thing that's going to happen is this strand of DNA here is actually going to unzip or open up into two. RNA will then come in, which is this red band here, and create pairs, bases that match the DNA, but in RNA. So it's a little different than DNA, if you remember, because A's will pair with U in RNA. A does not pair with T. But if there's a T in the DNA, it will still pair with A. But anytime you have an A in the DNA, it's going to now go with U. That's RNA. I know it's a little confusing. So the RNA is created that matches with the DNA. And this is all happening in the nucleus where the DNA is. So you're going to see the RNA is made, then the RNA and the DNA leave the nucleus. They're in the cytoplasm right now. Now the RNA is going to attach into the ribosome, which is what you see here. So this big brown thing is the ribosome. Now what's going on here is super, super important. These green things here are tRNA. The tRNA will read one codon, which is three bases, three mRNA bases. So this green guy, the tRNA, will read those three bases, which is a codon. Then it will match those bases with an amino acid, which would be like this, GLY, PRO, MET. Those are different amino acids that the tRNA is matching with the codons. Those amino acids will start to build up, and you'll see them start to become a protein. So the tRNA is reading different codons, getting a new amino acid to build onto the chain. So LEU was just added. Another LEU is going to be added. And you can see it starting to kind of form into a ball. So all the amino acids are becoming a big ball, which will become a protein. So now you have your lovely protein. And that is where we should be right now. So this is my favorite lesson to teach you guys. It's so much fun, and there's a lot of fun stuff to do in the classroom, but since we're not in the classroom, I'm going to have to try and take what the fun stuff is and put it online. So I'm going to start with this video clip from Into the Spiderverse, which is one of my favorite movies. So just watch it and enjoy. That's all you have to do.
Wow.
Is it too crazy?
No, man. Miles, I see exactly what you're doing there, man. Yeah. You know, me and your dad used to do this back in the day.
Stop lying.
It's true. Then he took on the cop thing, and I don't know. He's a good guy, just, you know what I'm saying. All right, come on, man. I gotta roll. Miles, let's go.
Alright, so I have a question now after the video. You might already know the answer, but that's even better. So what happened to Miles when the spider bit him? We see this crazy looking spider bite the kid named Miles, and then all this crazy stuff happens. So take a minute and think about what actually happened to Miles when the spider bit him. If we're to actually think scientifically about the answer to that question, the radioactive spider bit Miles, which then sends all this radioactive toxins, or bad things, into Miles's blood. The toxins actually mutated his DNA, and that gave Miles these abilities like super speed, super strength, and all these super senses. So he can hear things and sense things that are dangerous. So he gets all these new abilities from his mutated DNA. And that's the reason I show you this clip, is we're going to focus on mutations of your DNA. So why not start with a perfect example of mutations, which would be Spider-Man. So a mutation is just a change that occurs in your DNA sequence, either because of a mistake when the DNA is copying itself, or as a result of environmental factors like too much light or cigarette smoke, or for Spider-Man, it was a radioactive spider. So your DNA can mutate by accident when the cell is being copied, or because you smoke too much, you get bit by a radioactive spider, a lot of different things. Exposure to chemicals can mutate your DNA. So there's environmental factors, and then there's just mistakes in your cells. So I'm going to go over a few types of mutations, and I'm going to try and make it as easy as possible. So we're going to start with a normal gene, and a normal gene will just be this sentence, but I want you to imagine that each of these words is kind of like a codon almost, and it pairs with an amino acid. So as the sun set, the bird ate the worm. That's a normal gene. Now, one type of mutation that can occur is something called a point mutation. And a point mutation is just a change in a single base. So that would look like a substitution. As the sun set, the bird ate the worm. So we have this D is switched out with a G. So that is your substitution. And that will change the sentence, as you can tell, because a bird is different than a bird. And it's just one letter is being changed. So the substitution that we just saw is one base being changed. The G is swapped out for the D is swapped out for the G. And it can change the entire gene. If you really look at it, the gene is different, right? Because the bird ate the worm instead of the bird. But it could also go unnoticed. So a substitution can sometimes not be horrible and cause this life-changing mutation because it doesn't actually change the amino acid. So if we are to actually break it down, and let's say that the word bird codes for this specific codon, UAU, that's the mRNA. We take that mRNA onto the codon chart, so UAU will code for the amino acid TYR. Okay, so that's the amino acid that would be in the normal gene. But now we have the substitution, and now we have UAC. That is our normal codon now because of the mutation. On the codon chart, UAC also makes TYR. So they both make the same amino acid, which is why sometimes these kinds of mutations aren't actually that bad because they still have the same amino acid. It's not like if we switch from TYR to a totally new protein like PRO, which might change the gene and give you super strength. But this is the same exact amino acid, so there's really no change. So I hope that kind of helps you see why some substitutions can go unnoticed. They don't actually have to change the entire gene. Now we're going to talk about frame shift mutations. Okay, and frame shift mutations cause the entire DNA strand to shift over, which really changes the DNA completely, and that's where you see some really extreme mutations. Okay, so there's deletions. So the deletion here is as the sun set, ate the worm. So now we've deleted the entire word of bird. And when a deletion occurs, one base is deleted and the entire frame has to shift. So I'm going to show you what that means as best as I can. So let's say we delete one of these bases. Okay, so we're going to delete this G right here, because a mutation occurs and makes it disappear. Okay, now we have this big gap, this big space right here that needs to be filled. So what's going to happen is all the DNA to the right is going to shift down. So all of these are now going to shift over one. Okay, so all of them are going to move, which means that every single letter is going to move and they might not all match up perfectly. So I'm going to draw the new letters in green. So the A will shift down and become an A. Okay, that C will shift down and become a C. T shifts down and becomes a T. And same with that last T. Okay, so now we have all the DNA bases have shifted, and if you look, they actually don't really match up at all because C does not go with A, T does not go with C, and G does not go with T. So this deletion has caused this big old mistake right here, which can lead to some serious issues. If that gene was really important and now looks like this, there's going to be a lot of issues. So that's why deletions, and the other one I'm going to teach you are insertions. They can cause some serious problems with your genes because the whole gene has to shift. And the other type of mutation that's pretty similar is insertions. So our normal gene is as the sun set, the bird ate the worm. Now it's as the sun set, the ugly bird ate the worm. So we've added a base, and this again causes a frame shift. So I'm going to show you a quick video that probably explains this better than my drawings would. So this is what insertion looks like.
An insertion adds a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by one. Again, the resulting protein will likely be non-functional.
So those are the main types of mutations you need to know. Point mutations like substitution, and frame shift mutations like deletion and insertion. I'm going to put a link to the video you just watched at the bottom so you can review it if you need that. Okay, so just remember that we've learned how DNA becomes RNA, and then the RNA codes for specific amino acids. The whole point of that is those amino acids build up into a big protein like this. So if something goes wrong, if any of the DNA is changed, that changes the codon, and that changes the amino acid, and that means the whole protein could be totally different. And this can lead to a lot of health issues and lifelong problems. So that's mutations.